the pathway, this is our peripheral nervous system. We're looking specifically at the efferent pathway from the spinal cord, representing the central nervous system, all the way out to the target muscle. And remember, this is specifically muscle because we're working with voluntary action. So this is going to be innervating specifically our muscles, okay, and our skeletal muscles. And when we take a look at that somatic pathways, you'll see that the efferent pathway starts, okay, we've had our interneurons, we've got our efferent pathway starting at the ventral gray horn, which is going to contain all the motor neurons, which is going to comprise the cell body, and the axon projecting out the ventral root to the spinal nerve, back out all the way to its target muscle. So here on the muscle, we're going to see the sarcolemma, and that's going to contain, and that would consist of the neuromuscular junction. This is the bridge from the axon terminals of that motor neuron over to the muscle itself. And what you see that is involved, the neurotransmitter, is going to be acetylcholine. And we'll talk more about our neurotransmitters in just a bit. And you can see that the pathways leading to, for the most part, are going to be myelinated. And this pretty much describes our lower motor neuron. So if I were to ask you, the lower motor neurons located from ventral gray horn all the way to the muscle itself. And then we talked about the other neurons involved. So these are going to be the inner neurons. And looking at the inner neurons in the central nervous system, you can see that we have the cerebral cortex, specifically our primary motor cortex, which is the back portion of the frontal lobe. And this area is going to govern the upper motor neurons that innervate those areas of the body. So areas that are going to have the most motor neurons are going to be the areas that require the most fine motor control. So areas like the mouth, the hands, lips, tongue, etc., that have the biggest area. This was our motor homunculus that you saw. And those pathways will lead all the way back out over to the lower motor neurons. And one of the reflexes that you saw, that we talked about, is going to be the Babinski reflex. And that's one of the reflexes that you'll be doing in today's lab, where I mentioned that the lower motor neurons that is involved in the pathway for the reflex is simply spinal cord, especially in early babies. And they test that reflex by stroking the foot itself and watching for the foot to branch out. And that's the Babinski reflex. And why babies have that is because the myelination on that upper motor neuron from that area that innervates the area that innervates the feet will travel down, and you'll see that this area is actually not myelinated. So the pathway isn't as strong as it would be in adults. Okay. So with that, we'll take a look at our pathways. We talked about the local circuit neurons. Local circuit neurons are the ones that recruit other motor neurons to perform an even bigger action, and I'll talk about that in our reflexes. Upper motor neurons are from the motor cortex down to the lower motor neurons, and then you'll see the pathways that travel back and forth. The reflexes, the spinal reflexes, these are reflexes that take place from the muscle itself and back into the central nervous system and back to the muscle. So this is going to complete the loop for us when we look at the entire pathway. So stretch reflex, the first of the reflexes that we'll talk about is the stretch reflex. And these are going to contain the muscle spindles, which is going to be the receptors. These are going to contain the dendrites that will detect the stretch in the muscle. And they run parallel to the muscle fibers. So when we look at these receptors, these neurons, they wrap around the fibers themselves and they travel out. So their whole purpose is to detect stretch of the muscle. This reflex starts. What the stimulus is the stretch that's caused by the patellar hammer that bangs on that tendon, which creates this entire pull on the muscle itself. So this stretch reflex detects a stretch, and it's going to go back. The afferent pathway is going to travel back to the dorsal gray horn, back to the the dorsal root, back to the dorsal gray horn, and it will actually terminate right back on the motor neuron that innervates the quadriceps, the same muscle that contains the muscle spindles. So what started off as a stretch results in an excitation of that lower motor neuron. An excitation of that lower motor neuron results in a contraction. So what starts off with a stretch results in a contraction. So this is a form of negative feedback. What started off with a stretch results in a contraction. Now that's not the only piece. Simply because when we contract the quadriceps, we want to make sure that the other muscle, the antagonist muscle, is going to do the exact opposite reaction. Now think about this. If I had contraction of the quadriceps and contraction of the hamstrings, would we see flexion of the knee?
No.
Or would we see extension of the knee?
No, nothing.
Neither, because they both contradict. You have both push and, well, not push and pull, but you're pulling from both sides. So therefore, we want a contraction in the quadriceps and we want a relaxation of the hamstrings. So that same afferent pathway innervates an inner neuron. And this inner neuron is inhibitory and inhibits that lower motor neuron that targets that hamstring. So here you see it excited, we see relaxation of the hamstring, we see contraction of the quadriceps, and therefore what we're going to be able to see is extension. Okay? We'll see that kick. And when you guys are doing the reflex, you'll see when you test on yourself, bang on the patellar tendon, and you'll notice the exact same reflex that's taking place. And this happens very quickly. If you take a look at the path, that entire path is monosynaptic. So it happens faster before you're even aware of it. And that's something that you can test with each other too, is when somebody's performing that patellar tendon, try and see how fast, if you can catch yourself before you actually react. So that time that it takes for that sensory neuron to travel all the way back to the brain, the reflex will occur before you even know it. Hence why it's called a reflex. Okay. Did I say a question? Everybody good? Yeah, okay. All right, so the other portion is the Golgi tendon reflex. And in this case, what you're going to be able to see is that the receptors are located on the tendon. So it's in series with the quadriceps muscle itself. The whole purpose of this reflex arc is to prevent excessive contraction. So the stimulus in this case is contraction of the muscle that's placing a load on that tendon organ. And so what's going to happen is the neurons, the dendrites that are being targeted in the tendon organ are going to light up. It's going to send afferent information back the same pathway, but what that's going to do, it's going to target an inhibitory inner neuron. Because remember, we started off with a contraction. We want to now limit the contraction. So we're going to recruit an inhibitory inner neuron that will inhibit contraction of that quadriceps. So what is this inhibitory inner neuron doing? It's basically doing exactly what it's supposed to do. It's inhibiting. So here you can expect that we're going to result in hyperpolarization of that lower motor neuron to make it less likely to fire. Now, that antagonist muscle, the hamstring, we want to, if we're going to cause relaxation of the quadriceps, we want to create contraction of the hamstrings. So here you're going to see an excitatory inner neuron for the hamstrings that will target over here. So here you can clearly see the motor neurons are clearly located in two different areas. One that's involved for the quadriceps, the other one that's involved in the hamstrings itself. So the example that I used would be excessive muscle contraction. Take for instance, and a good example is always weightlifting. You place too much effort on a joint, and what's going to happen is it's going to limit the contraction. So at that point, you'll notice upon excessive load, the muscle fails. Okay. Another example would be stretching. Excessive stretching of the muscle will create, not only would you start to see the muscle spindles being stretched, which would start the contraction, but the Golgi tendon will override it. It'll detect way too much load on the muscle itself, and then it'll cause it to relax. And that's what kind of gives that flexibility in the muscle itself. It can accommodate modes, but to a certain extent. Does that help? Yes?
So for
the polysynaptic, yes. So this is considered monosynaptic. This is the muscle spindle, where the same neuron, the same sensory neuron, comes in and excites the lower motor neuron. There's only one synapse for the muscle spindle. Okay? At least specifically for the same agonist muscle. Now, this portion right here, which is inhibition of the antagonist muscle, as well as the Golgi tendon organ, require two synapses. And in this case, what we're looking at is because we have an inhibitory interneuron that's involved. And that inhibitory interneuron is inhibiting that lower motor neuron. Okay. So that's what separates it, is that you have the interneuron that comes in to allow inhibition or excitation of another muscle. Is that a little clearer? So in this case, if I can go over the monosynaptic, notice that this area, if I want to cause contraction of a muscle, all I need to do is excite that motor neuron, right? And that's what that sensory neuron is doing. It's coming in directly exciting that motor neuron. And there's only one pathway through. So one synapse in between the sensory neuron and the motor neuron. Why don't we see that with the other side? Well, our neuron is always going to be excitatory. Our sensory neuron is always going to be excitatory. So what we would want to do in order to inhibit that lower motor neuron, we would actually need to recruit something that would secrete inhibitory neurotransmitters. The sensory neuron can't do that. That's where the interneuron comes in. Now, this spinal reflex, this specific reflex is one in which we talked about. This is stubbing your toe. And in this reflex, what we're seeing is both a ipsilateral response, things that are happening on the same side of the stimulus, and things that are happening on the contralateral side. And so what I explained in this reflex is that stubbing of the toe. Here you can see the right leg. This person stubs their toe, and what they want to do is withdraw that foot away from the stimulus. And so this is going to involve the hamstrings. So here you'll see the response of the toe. You'll see the sensory neurons coming back in. It's going to innervate the inner neurons that will come in and target the hamstring. Okay. So what we're going to be able to see here with innervation of the hamstring, you're going to see our local circuit neurons that will travel to adjacent lower motor neurons. I'm sorry, adjacent inner neurons. And what that's doing, these are local circuit neurons that will recruit other motor neurons to create more of excitatory response on that hamstring. So this is going to create that exaggerated withdrawal response. So that's what's happening on the ipsilateral side. What's happening on the contralateral side, when stubbing that right toe, you're going to want to stabilize yourself by flexing the muscles on the contralateral leg. So what we're going to want to do is we're going to want to be able to recruit the quadriceps, fire the quadriceps to contract and stabilize. So this wouldn't be the only thing. You're also stabilizing your hips, you're stabilizing your core. There's a lot going on that contralateral side. But nonetheless, you would see that same interneuron that came in earlier. Same interneuron would travel to the other side to create excitation of the quadriceps on the contralateral leg. You'll see our local circuit neurons that come in and also innervate more of the quadriceps, creating contraction on that other side. Okay? So those local circuit neurons are not only the ones that are going to be recruiting the quadriceps, they'll come in, they'll also recruit the motor neurons for the hip, they'll recruit the motor neurons for the calves, and everything else to stabilize and balance. What questions do we have about that reflex? You guys are good? Okay. So with that, that describes everything that's happening with the somatic nervous system. Now we're going to take a look at the autonomic pathways. And the autonomic pathways are, as I mentioned in the very beginning of this lecture, is everything that is going to be autonomically regulated. Everything that's related to autonomic regulation of the visceral organs, glands, smooth muscle, you name it. Everything besides skeletal muscle. And as we mentioned in that car example, you saw certain activities, certain things that the person was not able to control. Right? We talked about the somatic responses, things like moving away from the car. Now, the autonomic pathways were responsible for the activities that the person had no control over, which you guys contributed as a rise in respiratory rate, rise in heart rate, and then all the other autonomic responses we'll talk about. So you'll see that at the end of today. So we're looking specifically at things that speed up glands and things that also slow things down. So we're going to be able to divide the autonomic nervous system into two pathways, the sympathetic and parasympathetic nervous system. And what we're going to be spending the rest of this lecture is taking a look at the differences between the two as part of the autonomic nervous system. So this we're going to be looking at both the afferent and efferent information. Afferent information that gives us, and this talks more about our sensory lecture that you guys will have in that video, is going to be sending information to the autonomic nervous system. Is there food in the stomach? Is digestion taking place? Are enzymes being secreted? Are hormones being released? Are we able to regulate blood pressure with spikes in blood pressure? If someone's CO2 in their blood increases, what is that? That information is going to contain afferent information that's going to the autonomic nervous system, and then the motor response is going to come back out the efferent pathways. So we'll take a look at the responses afterwards. Now the key difference with the autonomic nervous system compared to the somatic is that the autonomic pathways involve a two-neuron system. You saw in the somatic nervous system, you saw the motor neuron that starts from the spinal cord out directly to the muscle. In this case, autonomic is going to involve two. So what falls in between? What takes place in between is the autonomic ganglion that's present in between the first neuron and the second neuron. And the way we define our first neuron and the second neuron is we use the terms preganglion and postganglion. Preganglion from the spinal cord to the ganglion, and then from the ganglion to the target cell. Okay. And what exactly are going to be our targets? Either the smooth muscle, we're going to look at cardiac muscle, as well as any of the glands that we're going to be looking at that'll be able to secrete some of the hormones, look at some of the exocrine stuff that are going to secrete whatever we're trying to secrete, whether it's going to be sweat, whether it's going to be digestive enzymes, you name it. So where do we see the differences between the preganglion and postganglion? And again, I'll talk more about the details behind it, but one of the first things that you can tell that's one of the major differences, take a look at your preganglion neurons and your postganglion neurons. What you'll notice about the preganglion neurons is that they're myelinated. So you expect fast transmission from the spinal cord out to the ganglion. And now for our postganglion neuron, from the ganglion over to the target tissue, you'll notice are going to be unmyelinated. So this will be saltatory conduction. This will be continuous conduction. So expect this preganglion to be faster. Now, the main difference that we're going to be going over in our sympathetic and parasympathetic is going to be the location where that ganglion is located. So keep that in mind. That's going to be the big difference between the two. These little pieces right here, some glands are innervated directly by the preganglion neuron. That's okay. I'll talk about that towards the end, because while I'm describing a majority of the cases, the general rule behind these, there's always going to be exceptions to the rule. So looking at that, you're going to see where the origins, where do we expect to see the preganglion cell body? Our lower motor neurons, you know, are already located on the ventral gray horn. You were to see on here where this corresponds to. All of this would innervate skeletal muscle. Where do we expect to see the autonomic cell bodies? We are going to see them in the lateral gray horns. So if I were to ask you the difference between the two, if I were to pinpoint on a spinal cord section and say where would you expect this to innervate, you should know that these are autonomic pathways. So this should be our visceral organs. So let's take a look at that efferent pathway from the lateral gray horn out to the ganglion. So looking at this pathway, you can be able to see the same rami that we looked at earlier in today's lecture, in Thursday's lecture. And again, the rami, you'll see the dorsal rami, you'll see the ventral rami, and you'll see the rami communicantes. So what we learned in the dorsal rami and the ventral rami, these are going to be, the dorsal rami is the branch out of the spinal nerve that will innervate the muscles of the back. Ventral rami will innervate the muscles of the anterolateral region, everything in front of the spinal cord and to the side. And then the rami communicantes is going to be specifically for the autonomic pathway. And what you'll see is the lateral gray horn. You're going to see the cell bodies for the preganglion neurons. They will travel out from the lateral gray horn, they'll travel out the ventral root, they'll converge back onto the spinal nerve, and then they'll exit back out the rami communicantes. Now, where that ganglion is located, you're going to see there is a sympathetic ganglion, which I'll talk about in a little bit, that runs very close to the spinal cord itself. And I'll be able to show you one, because what we're looking at is a specific slice. I'll be able to kind of twist this three-dimensionally so that you can see what it looks like three-dimensional. For the most part, what you'll be able to see, the pathways that lead out for the sympathetic pathways will travel out the rest of the rami communicantes, while some will travel back out the ventral rami. So you'll see a little loop that comes back in and comes right back out. So now what we'd like to take a look at is to really compare and contrast visually between the sympathetic and parasympathetic division. Here you'll see sympathetic division to your left and parasympathetic to the right. And some of the differences that you can see, at least one of the main differences you can see, is where the preganglion neurons target. So what I want to point out for the sympathetic division is you can see that there is a sympathetic trunk ganglion. It's literally a cluster of cell bodies. It's about 25,000 cell bodies located on this trunk ganglion, and it runs parallel on both sides to the spine itself. And I'll show you another slide of what that looks like specifically. You can see for most of the neurons, you can see that the preganglion neurons travel from the spine out to the sympathetic trunk ganglion. And then you'll have your postganglion that travel directly to their target organs. Now, another thing you can note about the sympathetic division is that most of the nerves start from the thoracic over down to the lumbar. While looking at the parasympathetic, the nerves are really going to be the cranial nerves and the sacral nerves. So these are going to be where the preganglion neurons are located. So looking at that, we'll take a look more at the neurotransmitters a little bit later. That's really going to be involved in the preganglion versus postganglion. So it's clear to see where the ganglion are located. Again, sympathetic trunk ganglion. The question is, where are the ganglion for the parasympathetic? And I'll be able to show you that later. But they're located close to. You probably won't be able to see it. Let's see if I can actually zoom in. Might not be the best image. There, maybe if you can see the stomach. Here's the location of our preganglion neuron. And they come out all the way out to the stomach, and they travel all the way out through. And the ganglion is actually located right there on the stomach itself. Notice the differences in size. This is our preganglion neuron and our postganglion neuron. Again, more to come on that when we look at when we break down each of those divisions in just a little bit. The image to your left here is just really showing you the sympathetic trunk ganglion. And again, it runs parallel to, right down by the sides. That's all I had for that slide. So the green is reflecting the sympathetic trunk ganglion. So let's first start off with the parasympathetic division. So parasympathetic division, and I'll talk about in our later slide, I'll talk about what specific things are parasympathetic and which things are going to be sympathetic. So the parasympathetic division, just to give you kind of an anatomical orientation first, is called the craniosacral division. Why is that? Because we involve the cranial nerves and we involve the sacral nerves. These are going to be the locations of the cell bodies on the lateral gray horns of the sacral as well as the cervical slices. And they're going to travel all the way out. We have these very long preganglion axons that are going to travel to the ganglions that are located close to the target organ. As I mentioned, the stomach, here you can see the preganglion neuron running all the way up to the point you reach the stomach, and there's a whole bunch of cell bodies located right along the stomach. And if you want to take a look at what that looks like, there it is. It's a bunch of clusters right along the stomach itself. So we expect to see long preganglion axons that come in and target that ganglion for the parasympathetic division. Now, if we look at the postganglion neurons, they're going to start at that ganglia surrounding the tissue, surrounding the target organ, and they will terminate specifically on the smooth muscle or the gland themselves. Very short postganglion neurons. Now, the other ones where you're going to be able to see, so for the most part, like I mentioned, if you were to take a look at the digestive system, the ganglions are going to be located right all along the tissue. But if you were to look at the ones that originate from our cranial nerves, you're going to see that we have ganglion, ciliary ganglion. You'll see the pterygopalatine ganglion, submandibular ganglion, and the otic ganglion. And I'm not going to have you guys memorize that, but just recognize that the ganglion, again, are close to the target tissues. One thing I will point out that we will mention in our respiratory lecture as well as our cardiovascular lecture, take a look at cranial nerve 10, which is going to be our vagus nerve. Our vagus nerve is going to come in and innervate two of the things in our, they're going to innervate the heart, and they're going to innervate the lungs. So we'll talk about what parasympathetic pathways are going to do for those target tissues, especially when we get to those specific organ systems. Like I mentioned, ganglions located close to the target organ. So long preganglion axons, short postganglion axons. Now, looking at the sympathetic division is going to be called the thoracolumbar. Why is that? Because we're going to be involving the thoracic as well as the lumbar spinal cord segments. And where did we mention where the ganglions are located? Along the sympathetic trunk ganglion, which runs parallel to. So if we were to take a look at the preganglion neurons, we would expect the preganglions to be short. They travel from the spinal cord, again from the lateral great horn, out to the sympathetic trunk ganglion. You'll see point number 2 on here, prevertebral collateral ganglia. Where are these ganglia located? Well, the other ones are located, you'll see celiac, again, you don't need to memorize the names of these. You'll see the celiac ganglion, you'll see the superior mesenteric ganglion that target the intestines. You can see the inferior mesenteric ganglion that are going to innervate the bladder, the genitals, and the uterus. All of those 3 are not exactly close to the target organs, but they're closer to the spinal cords themselves. So why do we see ganglion outside of the sympathetic trunk ganglion? Take a look at the difference. Here you can see, and let me zoom in a little bit. If you were to take a look at where, let's take a look at T3. T3 comes in, innervates the sympathetic trunk ganglion, and targets the lungs. T4 does the same thing, innervates the preganglion neurons, terminate on the sympathetic trunk ganglion, and target the lungs. If you take a look at T5, T5 passes the sympathetic trunk ganglion, and targets the celiac ganglion. The postganglion neurons travel from the celiac ganglion over to the liver. So what's the difference between, or rather, let me phrase it this way, what's in between the lungs and the liver that would separate the sympathetic trunk ganglion from the collateral ganglion? The diaphragm. So here you're going to be able to see that on this slide. Here's a picture of the ganglia where you can see the sympathetic trunk ganglion, and that's going to be represented in yellow. And anything that travels out of the spinal cord will travel out and terminate on the sympathetic trunk ganglion. And that's everything from T4 and above. Anything below is going to involve the ganglion below the diaphragm, which you can see at the top of the screen. Here you can see our celiac, you can see our superior mesenteric, and you can see our inferior mesenteric. Again, close to the spinal cord, but they're going to be outside because the diaphragm is going to help separate everything from from the spinal cord out. You're going to see these are going to be where the collateral ganglia are located. Okay, so now anatomically you can see the main difference between these. So with that, like I mentioned, our postganglion neurons are going to travel the furthest. They're going from the sympathetic trunk ganglion or any of the other three ganglion collateral bodies that travel out. And they'll travel, the postganglion neurons will travel the longest distance over to the target organs. So that helps describe what's taking place, what's going to be described in the next couple slides. Our sympathetic trunk ganglion, which again travels off to the side. Here you can see the spinal cord traveling through. You can see the same pathways out here. You'll see this, the dorsal root, you'll see the ventral root. They come together and terminate right onto that sympathetic trunk ganglion itself. So here you can see the nice organization in the thorax itself. In the thoracic cavity, you can see our arteries, veins, nerve, and that nerve directly goes to that ganglion. Now, a couple things I wanted to mention, because I did mention a couple exceptions to the rule. And the two main ones that you're going to be able to see, let me start off with point number 2. Chromaffin cells of the adrenal medulla. So if we're going to take a look at the adrenal glands, right up on top of the kidneys, the little hats on top of the kidneys, you can see that the target specifically for the adrenal glands is going to come out between T9 and T10. So T9, the lateral gray horn on T9 is going to come out and bypass the sympathetic trunk ganglion. It's going to actually bypass the celiac ganglion, and it's going to travel directly to the adrenal gland. This is the preganglion neuron that bypasses 2 of these and innervates directly on the adrenal gland. So the length of the preganglion neuron is from T9 and T10 to the adrenal gland. Where exactly is the postganglion neuron? It's actually within the adrenal gland itself. So the adrenal glands are modified, specifically the medulla area is modified neurons that have turned into endocrine cells. In fact, they have both neuroendocrine functions. And anybody that's a specialist in neuroendocrinology works specifically with the sympathetic pathways innervating on the adrenal glands. Okay, so this is all related to the production of adrenaline, which we'll describe in just a little bit. So if you're to take a look at that pathway, it's a preganglion that terminates right into the adrenals. Now the other thing is to take a look at T5. T5 you can see on the other side, starts from the spine, travels to the sympathetic trunk ganglion. Our postganglion neurons travel directly to the sweat glands. And the sweat glands, the postganglion neurotransmitter is going to be acetylcholine. And again, I'll talk about the neurotransmitters in a bit. So given all that, both parasympathetic and sympathetic, I've been able to pinpoint the differences and where the locations of the preganglion cell bodies are located. You know where the ganglions are located. We've yet to talk about the neurotransmitters. What questions do you have about parasympathetic and sympathetic?
So you said the second one, the postganglion cells are located where? On the kidney? Of the chromaffin cells of adrenal medulla?
Yeah, so the cells themselves are located they're called chromaffin cells. They're in the adrenals. And if you take a look at the solid line represents the preganglion neurons, and it goes directly to the adrenals.
So the posts are located on the...
It's inside the adrenals. So they're modified postganglion neurons that are of endocrine functions. They're secreting neurotransmitters, but these neurotransmitters are going to be going into the circulatory system, which will make much more sense when we talk about the neurotransmitters later on. Everybody good? So now that you understand the direction and the orientation of parasympathetic and sympathetic, let's take a look at some of the sympathetic responses. Let's see if I have my container up. Give me a second. I'll get that loaded up. I have like 4 videos of sympathetic responses.
But it's still too small.
Okay, so I'm going to show you this video. This video is a video of someone recording right before a tornado. Okay, so this is a tornado somewhere out in the Midwest. You can see what's taking place before, and you can see what's taking place after. And just pay attention to the responses of everybody that are pretty much in sheer panic afterwards. So we can get this first one started. Anybody ever lived in the Midwest?
All right.
No? Lucky we don't have these.
Holy shit. All right, I gotta go. I'm coming, honey. Shit, where you at, honey? God damn, it's bad. All right, stay down, honey. Oh, shit, Josie. Uh-oh. It's we're gonna get some shit here, honey. I hope somebody. Oh my God! Okay, honey. Holy shit! Stay in. Oh, man. Oh. Shit!
Don't go out there.
No, not yet.
No.
Oh. Oh my God, Josie.
Notice the camera. Does it look like he's
Holy
holding the camera
moly.
steady?
No.
Oh my God. Our house is freaking destroyed, Josie. Oh my
They've
God.
got the, I think, like tornado shelters underground.
Where's Autumn? Oh. I've never seen anything like it. Oh, shit. I don't know. Let's go. We gotta see if anybody else's house is up. Josie, come on. Hang on a second. Oh, Jesus H. I'm in shock. I can't believe this happened. Oh my
Hear
God,
the
Josie.
breathing,
The neighbors' houses are gone.
hyperventilation.
Damn, I hope nobody's hurt.
You might not be able to hear it, but his heart is pounding. You can clearly see his shaking.
Come on, Josie, let's get out of here. The tornado's passed. We gotta get out of here and get to... Josie, come on now. I know it's scary. Come on now. And watch the nails. That's fine.
You can hear the horn in the background too.
Josie!
You saw this earlier. You saw the houses standing completely wiped out.
Holy shit! Josie, we gotta get out of here outside where it's safe, where the structure falls. That's alright. Fuck, there's a car right there. Right there! Alright, let's go. We gotta get to the neighbors.
That's a good one. Next one to look at is this guy right here. Yes, that is a selfie. You'll be able to see this in just a bit. This is a tornado chaser.
Right. I'm just past Narkarula Station and I've just been watching this Willy Foreman. Check that out, that is one of the big ones I've seen a few here.
No idea what he's
That
saying.
is absolute demonstration, it's right there.
hyperventilation. So like I said, you can hear the responses, things that he's not able to control. Did I have another one? Oh, this is the same one. So when you look at the responses, what typical responses would you see from somebody like this? Elevated heart rate, you start to see a rise in blood pressure, increased respiration rate. That stuff we've talked about. Why do we typically see this? Well, this is a response of fight or flight. And you've probably heard this from old school psychology classes. Sympathetic responses are fight or flight. And that's exactly what this guy is doing, is having the ability to run as fast as possible. And by provoking this sympathetic response, you can see a very exaggerated response in things. You may have heard of people being able to perform a lot better than they would have ever been able to. If you ever heard of the things that you may have heard of would be like a baby trapped under a car and a mother being able to lift a car off the baby. You'll see people able to, and something that I remember is running around UCLA campus. I used to run around the perimeter and being fairly tired running, but I remember a dog chasing me. And nothing got me to run faster than this huge dog chasing after me. All of a sudden you pick up like a second wind, and I've never ran as fast before. So things like that. And what is exactly happening in the sympathetic response is that you want to start to allocate more of your blood, more of the glucose, you want to send more oxygen over to muscles. You want to be able to pull away excess CO2 because these areas, the critical organs, your skeletal muscle, your lungs, your heart, and your brain are going to be working more. So therefore, you want to send more of your nutrients and more of those gases over to those areas. At the same time, we also want to be able to withdraw blood in areas that are non-critical. Areas such as the digestive system, reproductive, as well as the urinary organs. These areas don't need the blood that's allocated over to them. We'll learn this response as we go into those specific points. When we talk about the lungs respiratory lecture, you'll see what the sympathetic response is going to do specifically to the lungs. We'll see what it's going to be able to do to the heart, and not really talk so much about the brain. And then, taking a look at the same token, what's happening when we're pulling blood away. The term I'll be using is shunting blood away from those areas, from digestive, reproductive, and the urinary organs. Now the parasympathetic response, on the other hand, is the exact opposite. It's the rest and digest. So this is what's going to take place after, let's say, a sympathetic response. And I'll talk about that in just a little bit. But this is our way that we typically would see in a stress-free response. Let's say you're just laying on the couch. You'll typically see what other responses take place: salivation, lacrimation, urination, digestion, and defecation, especially with these right here where you can see rest and digest. Now the acronym that you can use, the mnemonic that you can use to remember this, is SLUDD: salivation, lacrimation, urination, digestion, and defecation. So these things take place when we don't have when we don't have a sympathetic response taking place. Also, what happens in the case of, think about that what happened after any sympathetic response. Okay, so typically what you would see after something scary happens, you may see the urge to urinate afterwards. You'll see lacrimation, the formation of tears. You can see people tear up, especially after a traumatic event, and something that they can't control. Why is that? That's part of the parasympathetic response. Parasympathetic response that takes place after something sympathetic. So sympathetic is going to ramp your system back up. Parasympathetic is going to bring you back down. And then, like I said, those two work hand in hand to speed up organs and to slow others down. So taking a look at these, here you can see examples of the sympathetic and parasympathetic response on the target tissues that we could be taking a look at. Things that are going to happen in the iris, salivary glands, what happens specifically in the heart, lung, stomach, etc. So one of the things that we'll be able to talk about in, at least that you'll be able to hear in our sensory lecture, we'll take a look at what happens with somebody who's having a sympathetic response. What would you expect to see? you're going to see pupil dilation. Why do we see that? Well, pupil dilation, when you open up the pupils, what you're going to be able to see is you're going to be able to get more light in. That's more sensory input being able to come in, especially in fight or flight situations. You're going to see heart rate increase and the heart contraction force increase. If you take a look at the lungs, what's happening is you see the bronchial muscles, and I'll show you this later on, where the bronchial muscles that surround the bronchioles that lead into the lungs will start to open up, and that allows more air to flow into the lungs. In the stomach, you'll see peristalsis, and we'll go over these terms later on, but these are things where we want to reduce the muscle motion of the stomach. Why? Because we're not, in fight or flight situations, we're not really focused on digestion, so we want to limit that. That we expect to see in parasympathetic, where you start to see increased motility, more movement of the stomach during rest and digest. Small intestine, same thing. Large intestine, less focus on the digestive system in sympathetic pathways, more focus on parasympathetic pathways. And next, what we want to do is to take a look and hone in back on the neurotransmitters. And the last 5 slides will sum up what we're going to be talking about by just looking at the neurotransmitters. By now we've talked about the neurons, we've talked about where the cell bodies of those neurons are located for our preganglion or our postganglion neurons. We've talked about where those ganglions are located. Now we have to talk about the neurotransmitters that are secreted. And the main gist of all of these, because these are involving a 2-neuron system, we expect to see 2 types of neurotransmitters. So the first we're going to see are cholinergic neurons. And if you take a look at the term cholinergic, you'll see there's going to involve acetylcholine. That means these receptors, these neurons are going to be able to secrete acetylcholine. And the general rule behind all of these is that both, and if I can draw preganglion for both, you know what, I've got that on the next slide over. And I'll just show you these. Here we're going to take a look at our parasympathetic pathways. Parasympathetic division, we're going to see our cell bodies that are located in the lateral gray horns in the thoracic these are going to be the neurons that are located on the lateral gray horns that will come in and terminate on the ganglions close to the target cell. Like I said earlier, if you take a look at each one of these, you can see that the preganglion neurons are myelinated. Now, with our parasympathetic and our sympathetic pathways, our preganglion neurons for sympathetic and parasympathetic will secrete acetylcholine. Okay, so all preganglion neurons will secrete acetylcholine. The postganglion neurons for our parasympathetic pathways will also secrete acetylcholine. Now that talks about the acetylcholine. Now we're going to need to talk about, in this case, we'll talk about the receptors involved. So the receptors that are located, that pick up acetylcholine, you're going to be able to see that the receptors are named differently. And they're named differently based on their ability to not only bind to acetylcholine, but to the other agonists that bind to them. So you'll see that the acetylcholine binds to nicotinic receptors located on the dendrites of the postganglion neuron. And they're called nicotinic receptors because not only do they respond to acetylcholine, they also respond to nicotine. So nicotine, as you know, is something that that is excitatory. It comes in, it's the same nicotine that's found in tobacco, but it comes in and excites all of our preganglion neurons. So nicotinic receptors located on the postganglion dendrites, and then if we take a look at the target cells are going to contain muscarinic receptors that also bind to acetylcholine. Why are they muscarinic? It's because they can also bind to another chemical called muscarine, which is derived from a specific type of mushroom. And in all of these pathways, whether they're the nicotinic receptors or the muscarinic receptors, they're all going to result in depolarization. And typically with those are going to involve the movement of sodium into the next target tissue, whether it's going to be the postganglion or the target tissue itself. Now the exception to that is going to be our sympathetic pathways, specifically from our postganglion neuron to the target tissue. And if you were to take a look at, again, our sympathetic pathways, you'll see nicotinic receptors located on the postganglion neuron, but the postganglion neuron is going to secrete adrenaline. Here you can see adrenaline or norepinephrine. So these are going to be adrenergic. And here you can see adrenergic neurons, which secrete epinephrine or norepinephrine. So these adrenergic neurons are called our catecholamines. Where epinephrine is also known as adrenaline, and norepinephrine is also known as norepinephrine, or I'm sorry, noradrenaline. So in almost all cases, our sympathetic postganglion neurons will be secreting epinephrine or norepinephrine. Now the receptors for epinephrine or norepinephrine, you're going to see we have 5 different types of receptors. Why do we have so many different types of receptors? Well, like I said earlier, some of the receptors will involve excitation of the target organ, and some will involve inhibition of the target organ. In other words, some things will start to get turned on, and some things will get turned off. So as I showed you earlier, for sympathetic pathways, you'll be able to see where we'll see excitation of the heart. We'll also see inhibition of the organs in the digestive system. So some things turn on and some things turn off. Now the other exception to the rule, and as I mentioned earlier when we looked at T5 that innervates that comes in and innervates the sympathetic trunk ganglion and reaches the the sweat glands. The sweat glands in these cases are going to be utilizing acetylcholine. So here in every pathway outside of this area, everything from the postganglion neuron is going to be sending out epinephrine to all these target tissues, not the sweat glands. So now that we've described that, let's take a look at some of the adrenergic receptors. And we'll be, again, looking at these for the next couple slides. Adrenergic receptors are going to be divided up into 5 different types of receptors, of which we have an alpha and a beta class. And no, I'm not going to ask you really the main differences between the alpha and beta classes, so I'm not going to require that of you. But we will look at some particular examples on the next couple slides, especially those that have a clinical application. So again, in none of these receptors I'm going to have you memorize which one bind to norepinephrine or epinephrine, because really all these receptors differ whether they bind only epinephrine, or a combination of both. Now what's interesting about epinephrine is, like I said, it is secreted by postganglion neurons, but we also produce epinephrine in the adrenals. Again, the adrenals are the modified postganglion neurons. That epinephrine is something that makes its way through the circulatory system so they can go in and target those tissues as well. So not only is it a neurotransmitter in this case, it's also going to be a hormone that's produced by adrenals that can make more of a systemic response. Something that we'll describe after our sensory lecture where we go into the hormone effect rather than the direct connection that we see with our nerve pathways. And the main idea behind adrenergic receptors, and I'm just going to draw the cell membrane. And on the cell membrane, we're going to have a receptor. And this is going to be a receptor for epinephrine. So epi is going to bind to this receptor, and it's going to trigger excitation. And this is where you're going to be able to see the, and I believe I described this earlier, is the G proteins. G proteins that are involved. What's going to take place with the receptor is the receptor is going to turn on. So this is the receptor. This is our G protein. And I'll be drawing this numerous times. You'll be able to see this in our, I have slides that go into more detail on this in our hormone lecture. That G protein comes in and turns on an enzyme called adenylate cyclase. And adenylate cyclase, when turned on by the G proteins after binding to epinephrine, will start to produce, will turn ATP into cyclic AMP. And the cyclic AMP is going to be this internal signal that's going to come in and have some direct effect on the cell itself. Whether that's going to be increased production of proteins within the DNA. Maybe that cyclic AMP will come in and turn on other enzymes, and so forth. So that's what's going to take place. And again, we'll go into more detail about that later. So looking at that, we're going to take a look at some of the receptors, so some of the adrenergic receptors on different areas of the body. But from this perspective, what we're going to be looking at are the 5 types: A1, A2, B1, B2, and B3. So looking at these receptors, here you can see A1, A2, again, the 5 different types of receptors. Let's take a look at beta 2. So beta 2 comes in and innervates. Where do we typically see beta 2 receptors?
Airways.
Yeah, exactly. I was trying to zoom in. And there we go. Beta 2. Smooth muscles in the walls of the airways. And then I'm not going to talk about the blood vessels and hearts, but we'll talk about that later. And what we would expect with the smooth muscles in the airways, when epinephrine binds to beta 2 receptors in those located in the smooth muscle walls of the airways. So I'm just going to draw that out. So here's the bronchioles that lead into the lungs, and you have smooth muscle wrapping around it. What we want to do is increase air into that area. So we want to relax those smooth muscles to allow more air to go in. So with epinephrine coming in, we would see that beta 2 would result, stimulation of that beta 2 receptor would involve inhibition of those airways. So relaxation of the smooth muscle airways. So naturally what we want to do, we may not want to shoot epinephrine into into somebody. Well, it depends. We may need to. In the case of somebody that needs an EpiPen after an anaphylactic shock, if somebody's having difficulty in breathing due to an allergic response, anaphylactic response, they would typically get epinephrine, because epinephrine is going to come in, it's going to bind to the smooth muscles in the airways, it's going to cause relaxation, causing the airways to expand. Otherwise, if somebody is having, let's say, like an asthmatic response, where they're having difficulty breathing, what you can do is you can use something that's similar to epinephrine, something called an agonist. And that agonist that we have is an example called albuterol. Does anybody here use albuterol at all? Yeah. So albuterol works specifically on the smooth muscle airways by causing relaxation of that smooth muscle. And it works fairly fast, and that's why it's called an emergency medication, because it usually works in about 10, 15 seconds, and really takes the strongest effect after 2 minutes. And once you relax the airways, somebody can breathe a lot better than what they did before. So that's an agonist, meaning it's going to bind to the beta 2 receptors and do exactly what epinephrine does, to open up the airways. Now let's take a look at another example where I'm going to take a look at beta 1. And beta 1 Where we would see beta-1 receptors, we would see them in the cardiac muscle fibers. And not only that, you're also going to see them in the kidneys that are going to be responsible for blood pressure. So cardiac muscle fibers, if I were to take epinephrine, they would come in and excite the beta-1 receptors located on the heart muscle fibers and cause excitation, meaning it's going to increase the rate of contraction. Okay. And that rate of contraction is going to increase. So if we're increasing the rate of contraction and increasing the contractile force, that's going to cause heart rate to increase. And people that have issues with blood pressure, we don't want them to have a faster heart rate. We want to slow it down to bring their blood pressure down. So clearly somebody that has hypertension, you don't want epinephrine binding to, because that would cause increased heart rate response. What we want to do is give them something that blocks those beta-1 receptors. Hence we call beta blockers. What beta blockers do is they come in and they bind to the receptors and compete with epinephrine that's supposed to bind, just like you remember from our first week's worth of lecture. They come in and compete and allow less epinephrine to bind, therefore decreasing the response. I'm pointing the wrong finger. Decrease the response of increased heart contraction and rate of contraction. So beta blockers or beta-1 antagonist slows down the heart rate by blocking epinephrine on cardiac muscle fibers. Another thing to point out, again, taking a look at some of the other health supplements out there, there are typical fat burner drinks that you've probably seen around, and any of those, and not just drinks, but other supplements as well. Some of the things that you might notice about those, and I know those are popular, I think they're still popular, I think people still take it. But there are some that work on the same adrenergic receptors. Any of those supplements that you take, you notice probably increase your heart rate quite a bit. Okay? They increase your blood pressure quite a bit, because that's exactly what they do, is they come in and stimulate those areas. And typically those areas that are targeted are the ones that bind to the beta-1 and beta-3. Beta-1 receptors not only are the ones located for causing excitation of the heart, but you also see the beta-1 receptors located on adipose cells, fat tissue. And epinephrine, when it binds to, or any other agonist that binds to the receptors on adipose cells, results in the breakdown of triglycerides. Basically the breakdown of fat to release fatty acids. Because fatty acids are going to be but they're going to be an energy source that is readily available. So those types of supplements want to target beta-1 receptors, which will also increase your heart rate. And they also will target beta-3 receptors, because these receptors are located on brown adipose tissue that are responsible for the production of heat. So there's something that we don't really go into, but brown adipose tissue. There's white, which is fat insulating. Brown adipose tissue is the one that's used readily for energy. So typically with those types of things, you'll see agonists that come in and will also cause a whole host of side effects in areas that are also excited with those types of receptors. Yes?
So can you take an agonist with an antagonist?
Then they would just cancel each other out. So if an agonist comes in, it can mimic the response, but an antagonist will come in and compete.
So for example, if you took albuterol, but you have a weak heart, and you took an antagonist, would that decrease the bronchodilation as well?
So looking at beta-2, but beta-2 really is mostly in the smooth muscles of airways. You'll see it's also in the blood vessels, but not really in the heart itself. So if you had epinephrine that was targeting the beta-2 receptors, that would work in terms of that would work specifically in the heart. The other receptors are going to be different for the beta-1. Okay, so if you were to take things that target the same, so if you had something, a beta-1 agonist and a beta-2 agonist, they just come in and compete for those same receptors. Okay, it just depends on which ones we're targeting specifically. So that ties in our pharmaceutical applications. The last one is to take a look at this. It's mostly text heavy. It goes in and shows you the somatic nervous system and the autonomic nervous system helps you compare and contrast these two. And again, looking at where the sensory input is, looking at the motor neuron pathway, of which you know, the somatic nervous system is 1 neuron pathway. Our autonomic is going to be a 2 neuron pathway. The neurotransmitters that are involved, their target cells, and then the responses that take place afterwards. So somatic resulting in contraction of skeletal muscle, and autonomic resulting in contraction, but not only contraction, but relaxation of smooth muscles, depending on where those smooth muscles are located, whether they're located in the heart, located in the digestive system. Some things get turned on, other things get turned off. And then this just gives you a visual summary between those, where somatic pathway represents that on the left, and the autonomic pathways for the 3 pathways on the right. The 2 pathways of which we talked about, parasympathetic pathway and the sympathetic pathway. And then this is going to be the adrenal pathway, which is going to be the exception, where these cells are preganglion neurons innervate the adrenals specifically to send epinephrine through the circulatory system. So really take a look at these 3 and being able to differentiate these between the 2. And that's really it. All right. So with that, that just ends today's. What I'm gonna have you guys do next is